Good morning or good afternoon. Hey guys, and welcome to the next little bit on population genetics. I'm going to start off today with a quick review of the stuff that we've been learning about in Mendel because it's absolutely vital for us to understand that before we move on. And then we're going to go ahead and talk a little bit about how all of the Mendelian genetics and inheritance patterns and all the stuff that we've been studying are going to lead into wide-scale big changes within a population. So without any further ado, let's go ahead and dive in. So just as a review for inheritance, we've discussed already that all of our genes, which are basically segments of DNA that code for a protein, are located on our chromosomes, which are individual strands of DNA. The typical human has 23 pairs of chromosomes, meaning they have 46 in total, and each one of those pairs is made up of a homologous pair, which is one chromosome inherited from the mom, one from the dad. Both chromosomes in each pair are going to be identical in terms of the genes that they possess and the order in which they possess them, but of course they are going to have a potential to have different alleles. So in other words, the dominant or recessive versions from either mom or dad. So when you inherit your chromosomes, within these we're going to have these little gene loci or locuses. These are going to be where the actual genes or segments of the DNA are located. In between those are non-coding regions of DNA that code for either different genes and things, or it could be that they code for transcription factors and stuff that help us to control which genes are cut on and off. And we'll talk more about that later on, but it's kind of like your epigenome, if you remember the stuff on epigenetics. Okay, so that goes ahead and covers that. And we talked about Mendelian inheritance. We talked about the law of segregation, wherein all of these organisms inherit just one copy of the gene from each parent, so you become a mix of your mom and your dad. You don't inherit, two chromosomes for one pair from your mom and two from your dad. You inherit one from mom, one from dad. So you're a great kind of shuffle of the two genetic makeups. And then there's a law of independent assortment, which means that your alleles that you inherit are independent of other ones. So it's not more or less likely, so long as, again, the genes are unlinked, it's not more or less unlikely or likely that you'll receive a certain allele if you receive one. Of course, that means if they're linked, that is, of course, not the case. And if the genes are linked, then you're going to have to inherit one with the other. So we talked about monohybrid crosses, Punnett squares, dihybrid crosses. Here's our monohybrid cross. Here's the Mendelian ratio when we take two hybrids, which we can also call a heterozygote. They have one copy of the dominant and one copy of the recessive. When we take two of those and we mix them together, we see a 3-to-1 dominant to recessive phenotype. In other words, these three individuals have at least one copy of the dominant allele and they show the dominant trait, versus this one individual who has the recessive allele, because they have two copies of the recessive allele and they show the recessive trait. So this is the typical monohybrid cross, the 3-to-1 golden ratio for this, and then this is with a single trait of interest. And of course we can also do this with the dihybrid. Basically takes the genotype or the genes of an individual and we go ahead and we foil them to see all the possible combinations or gametes that that person could make. Remember, gametes are sex cells, so these are all the possible sperm or egg special shuffled combinations they could make. And then we go ahead and we cross those in something called a dihybrid cross, assuming that they're unlinked, and we end up getting a 9-to-3 to 3-to-1 ratio of dominant dominant to dominant recessive to recessive dominant to recessive recessive. And so these are two separate traits. We can see the law of independent assortment is at work. We see the 9-to-3 to 3-to-1 ratio, all very Mendelian. Good. Then we talked about the fact that you can have patterns that differ from Mendel, right? So we have the linkage group, which is going to be where we have on one chromosome you have capital A, capital B. On the other one you have lowercase a, lowercase b. And you basically have to inherit them together. So when we look at a linked, Thomas Morgan was the guy that figured this out, and he did this with flies, whatever. Okay, so basically what it means is that when you're linked, you can only inherit the A and the D together, the big A, big D, or you can only inherit the little a, little d together. You can't inherit big A, little d, or or big D, little a, or any of that sort of thing. And if we look over here, though, if they're unlinked, you can do that foil thing that we looked at with the dihybrid, and you could get 4 possible gamete combinations, meaning that your offspring could be a lot more diverse. Over here, you're going to look just like your parents, one or the other. Over here, you're going to be shuffled up and look different from your parents, right? And so here, this is one of the ways in which we can destroy that Mendelian ratio. This is just showing you in recombination during prophase 1 of meiosis. If you guys remember, this is where we have crossing over happen. So we have our two bivalents, which basically means we have the two copies, the exact copies or sister chromatids are attached at the at the middle by a centromere. And so these bivalents right here are going to line up during prophase 1. They're going to cross over their genetic information. This is called a chiasmata, this little bridge that they form. And then you can see that they've swapped some of their alleles, and this just helps to create recombinant offspring. And recombinant offspring are going to be offspring that look different than the parents, and this is of course to increase the genetic diversity in this germ line, this gene line. And we can see that that worked. Now with a pair of linked genes, we would not have these recombinants, or the recombinants would happen much less frequently than the normals. And so, and we call the normals because they look like the parents. So in other words, these are new and novel combinations of the parents' alleles that they don't possess themselves. And if the recombinant number is high, then that means that the gene linkage is low. If the recombinant number is low, gene linkage is high. So in other words, if crossing over happens, the genes are probably unlinked, or they're not very closely linked. If a lot of crossing over happens, that means they're basically unlinked. If no crossing over occurs, that means they're completely linked. And then if just a little bit of crossing over occurs, that means that they're linked, but maybe not completely. Basically, gene linkage has to do with the distance between alleles. So if the alleles are super close together, they're more likely to be inherited together. If they're super far apart, they're more likely to be swapped when crossing over happens. So in this way, we can kind of see the distance between genes on a chromosome. So we can identify linkage when we look for the number of recombinant phenotypes. Again, we just go ahead and take the number of recombinant phenotypes. We take a look at them. Do we see that they are more or less than the parents, right? And we kind of decide that way. So here we can see that these guys represent these parents, and these guys represent this parent. And their numbers are fairly equal, and then the number of recombinants are fairly equal. So right here, this would be unlinked, right? For linked, we would go ahead and see that most of them would resemble either this parent or this parent, and very, very few of them, less than half, right, would represent recombinant. So in this case, we would see a linked phenotype or genotype. Okay, and then we have polygenic inheritance, which we talked about. Basically means multiple genes are going to code for a single thing. So this could be like your skin color or human weight or whatever. All these can be controlled by more than one gene. Monogenic traits are what Mendel was very lucky to work with, but a lot of traits are not monogenic. This, of course, means that they're controlled by one gene. Polygenic inheritance, it can be a spectrum, right? And so that's why people come in all different shapes and sizes and all sorts of colors and stuff. And this is just more examples of polygenic traits. So that's what we went over. So I just wanted to briefly go over that so we can understand. And then we went over chi-square. So we talked about chi-square in that wonderful example of HH and my provide us, and we basically said that we could use statistical methods of analysis to show that certain genotypes or certain traits could be more advantageous than others. Of course, we know this from evolution. We know that there are certain traits that can increase your reproductive fitness and your survival in the population, and those are more likely to be selected for. Now, that was an interesting thing, right, with the sickle cell, because sickle cell is typically something we want to try to avoid because it's deadly and dangerous for us. But having just one copy of that sickle cell gene gave us a sickle cell trait, which means that we would be immune to malaria. Or it would be a lot harder for malaria to kill us, and so it's kind of a defensive mechanism. And so nature was sort of using one, two wrongs don't make a right, typically, but it was using like one to kind of offset the other, right? And so we were kind of taking a look at those balances and seeing what happened in the population. So that's population genetics. We take the ideas of Mendelian and non-Mendelian inheritance patterns and we apply them to this ginormous population, and we see whether or not those trends come out. And that's basically what we've been looking at this whole time. So now we're going to go ahead and take that and we're going to apply that to that huge evolutionary concept. And we're going to take a look at how these inheritance patterns affect not only the adaptive changes within a population, but also can create speciation. So an entirely new species where they weren't there before. So let's go ahead and dive in. Okay, so we know already that the best definition of evolution, which is not quite here, the best definition of evolution we know is a change in the population, a change in the allele frequencies of a population over time. And so basically that just means which alleles are more common or less common within the population, and that's really dependent on whether they're beneficial or not beneficial, or just whether kind of luck of the draw. And we're going to look at various different ways that they can be changed, those allele frequencies, but that's basically what we're looking at. Now, I just want to point out that common or not common does not mean dominant or recessive. That is a huge misunderstanding. Remember that dominant and recessive just has to do with which protein is expressed. But dominant is not more common, and recessive is not less common. It depends on the population. If there are more recessive alleles within one population, the recessive is going to be more common. If there's more dominant in one population, the dominant will be more common. And this is really easy to kind of work through in your head. You can look it up online and stuff, or I can do a different video on it. But just remember, dominant and recessive, they're not common or uncommon. They just refer to, I guess really you could think about the shape of the proteins in the end. That determines some of the traits. Anyway, so we know what evolution is. We know that we're able to pass on our genes, that they're inheritable, that you can get traits from your mom and dad. And so again, we're going to look at the allele frequency. Now, when we're talking about the allele frequency in a population, because we're talking about so many different organisms, what we actually do, because we're looking at it through the lens of a geneticist, we kind of pretend that that population isn't made out of individuals, but it's just made out of a giant pool of genes. So in other words, we kind of get rid of all of the imaginings of like a herd of animals, and all we see is just this giant pool of big A's, little b's, all that sort of stuff. And that's what we measure, because we're looking for allele frequencies. Individuals, in this case, from this perspective, don't really matter. The individuals are just like containers that carry the genes. We don't really care that much about the containers. We care mostly about how the genes are interacting with each other, which ones are being inherited together, being passed on together, and which ones are surviving and which ones are not. Of course, the environment is acting upon the phenotype of the individual organisms, but in this case, we don't really care about that. Okay, so we're going to take a look at the gene pool overall. So the gene pool, if you see here, this is the total amount of the gene pool. So we wouldn't see mice, we would see CC, BB, AA, etc. Your large gene pools are going to typically be more diverse. And this just makes sense, right? Because you have more individuals, more opportunities for different alleles, and more opportunities for different individuals to mate with one another. If you look over here at the right-hand population, these individuals that want to mate, say this one wants to mate with someone else, well, they really don't have very many options in terms of the alleles that are available. Over here, if this person wants to mate with someone else, they have a lot of options in terms of what's available and a lot of potential combinations. So the larger the gene pool, typically the more diversity. Of course, this cannot always be the case, because if you think about like endangered species that we've done special breeding programs with, I mean, their gene pools are very, very small because they're working with a small amount of genetic diversity to start with. We'll talk about that in a little bit. Anyway, so gene pool compositions in terms of the letters of the alleles, dominant, recessive, and which alleles are present, changes over time, and that's really what we're talking about with evolution. Now, this could be genetic drift, which we'll talk about in a second, or natural selection, which you should remember. Okay, so when we talk about changes in allele frequencies, we talk about them in terms of a percentage or as a ratio. Now, typically, when you look in statistics, we don't usually always go from 0 to 100%. We can also talk about it from 0 to 1. 0 meaning that it's not present at all, and 1 that it's completely present or fixed in the population. So that's something to keep in mind in terms of notation and nomenclature. And your changes in allele frequency are going to reflect different things. Okay, it could either reflect that there's random processes like random organisms dying, natural disasters, stuff that's uncontrollable by nature and not controlled by natural selection, and that's what we call genetic drift. It's basically just the random stuff that happens every day that we can't control that changes the allele frequencies in the population. Then we have differential processes like natural selection, and differential means that we have some sort of directional or a change based off of some underlying cause. Okay, so for instance here, if I was talking about natural selection, I would expect to see if one of these allele frequencies was rising steadily and was just going in a single direction, I would probably say that's natural selection. If it was wobbling all over the place or went up suddenly or went down suddenly, I might think, oh, maybe that's just random processes like genetic drift. But if there's a continuous directional change, or if I see it changing in conjunction with something in the environment, that's more likely natural selection. So population bottlenecks, which we'll discuss in a moment, and the founder effect, will increase the rate of change that happens. So these two guys that work together, basically random processes are always happening because life happens. And natural selection is always happening. Sometimes they're at odds, sometimes they work together, and this makes it so that populations' gene pools are very variable in terms of their allele frequencies. So let's talk a little bit about some of the ways in which these allele frequencies can change. So genetic drift is one of the most common and most important ones. The idea of genetic drift, if you guys remember, is that we have individuals that are moving between populations and they're breeding between those populations. So if we think about it in terms of gene pool, basically some of these alleles are being poured out from one pool into the next, and then they mix with that pool. So if we see here, we see the green bugs and the brown bugs are all together here. And we see here that somebody's shoe comes down and crunches some of the bugs. And then we see, there's only brown bugs left. Okay, so in this case, if these green bugs had continued to interbreed with these other ones, we would probably see more green bugs mix in with the population. But because this person came down and squished all the green bugs, we see none of those green bugs moving around to each of the individual populations. And so what we see here is that this population will now remain brown. So we basically eliminated one of the alleles from the population and made it so that only one color can kind of go around. So this is one of those instances of a random occurrence that can totally crush or destroy something. Okay, so genetic drift. So it's usually greater when the population is smaller. So you can see because there are only 3 green bugs, they're really easy to squish. That means that it's very easy for us to change the allele frequency of this population. I mean, originally, if we had 1, 2, 3, 4, 5, 6, 7 bugs and 3 of them were green, that's like, what, just a little bit under 50% were green. But now, in one moment, we have gone from that to being 100% brown, whereas before we were just about 50% brown. So with the small population like this, it's very easy to change those allele frequencies very fast using a natural disaster or random accident. But genetic drift can also be enhanced by things like the population bottlenecks or the founder effect, which we'll talk about in a second. So genetic drift here's how I want you to think about it, because I think for a brief second I got this confused with gene flow in my head. So genetic drift is just random changes that occur to a population to change its allele frequencies. These can be like persons stepping on bugs, random deaths, whatever, but this is all genetic drift. It can also include population bottlenecks, which are natural disasters, or organisms moving around from one area to the next. So a population bottleneck is basically when we have a massive natural disaster that destroys at least 50% of the population. And so we can think of this as being a tsunami, an earthquake, a landslide, whatever. And so these are they could also be included by humans, right? Because humans can overhunt or drive certain organisms extinct. So the surviving population that's left is very, very small, and it has very little genetic variability because it represents a very small portion of the original variability of the original population. So we can see here we have green and blue and yellow and pink and orange. And down here, all the blues got destroyed in the natural disaster, right? And we only have pink and green and orange, and that's it. No more yellows or blues. They've been totally destroyed, and now we only have this. So they represent less genetic diversity. So this is an example of genetic drift. And then we have the founder effect, which is basically, you have a couple of individuals from one population that are like, we want to move, and so they move away from the main population, and they establish somewhere else, and we can see, wow, okay, when they do that, okay, there's a lot more of the orange here than there is the purple, and so you can imagine that this population here is going to probably be like mainly orange, whereas over here it might be around 50-50 orange and purple. So again, this is an example of genetic drift. It's much more effective with a smaller population, of course, because there's less genetic diversity, and it can lead to certain disorders that might have been present in the original members. So if we think about, there's an Amish population that has a high incidence of polydactyly, which means they have more than 5 fingers, or they have a weird number of fingers or toes. And that's because the individuals that left had a higher genetic predisposition for it, and because most of the population was just them for a long time, and they kind of interbred that way, that means that the frequency of that allele rose highly in that population that fragmented. Okay, and here's natural selection. We already know this. So natural selection, right, is if there's a certain phenotype that is selected against by the environment, like the green bugs are easier to see than the brown, then the environment will come in, select against those, and their frequency will decline the population. I don't think I have to go into too much detail with that, because that should be self-evident at this point. Okay, so there are different types of selection that we've discussed, and I'll go over these briefly. And these are patterns or ways in which we could kind of take a look at how the gene pool is changing in terms of its phenotype over time. The stabilizing is that the intermediate phenotype is favored over the extremes. So in other words, if we had small beaks, medium beaks, and large beaks, the medium beaks would be preferred in stabilizing selection. Directional means one extreme phenotype is selected at the expense of another. So in other words, beaks are getting larger all the time, or beaks are getting smaller all the time, but they're going in one direction. So the population is either increasing as a whole or declining as a whole. Now, does that mean that every individual is going to have grow a larger beak over their lifetime? No, it means that the individuals with larger beaks, if they're being selected for, are going to be better at reproducing and passing it on to the next generation than the ones with smaller beaks. And so we're going to see that change. Disruptive is going to be where both extremes are selected over the intermediate. So in this case, a big or small beak might be selected, but not a medium beak. Those are going to decline, and those alleles that cause medium beaks are going to decline in the population. So these are all really, these are describing allele frequencies, but they're operating off of the phenotype of the organisms here. You'll notice that natural selection is going to operate off the phenotypes a lot more than genetic drift. Genetic drift doesn't care about the phenotypes. It's just random occurrences. There, it's more useful to look at the alleles themselves. So here we see stabilizing selection. So we see the intermediate is favored, and there's different examples for that. We see here directional selection. So this is the original population. We see here that we've moved to being a darker color of mouse, probably to be able to hide ourselves better in the environment. And then we have disruptive. So maybe this means that there are light-colored plateaus and dark-colored plateaus, but not any plateaus that are this color. And so these two different colors of mice thrive, right? And we saw that also with the moth populations in bee Betularia. Okay, so here's one of the most important ideas of evolution, reproductive isolation. So when we isolate two different populations, essentially what occurs is that there's no gene flow between them. And gene flow is what I was trying to describe earlier. I got it confused in my head with genetic drift. Gene flow is where you have individuals that are kind of being poured from one population into the next. So they're basically, if you think about this gene pools, the alleles are moving from one population into the other. They're mixing together with that population and kind of injecting a little bit more variety into it. So reproductive isolation is when there is none of that. The pools are distinct from each other. There's no mixing, and they stay exactly as they are. Now, when this happens, we know that mutations are accruing constantly within gene pools. New mutations are arising all the time. New alleles can be forming through these processes of mutation, and so over time, basically, the pools are going to become more and more different from each other. You can imagine it as being, let's say that there's a pond up in a mountain. Like a clear, pristine mountain, where there's not a lot of pollution or anything or people. And then let's say that there's a pond that's sort of near to it, but near to a human settlement where people are around and maybe a factory nearby or something. And originally the two ponds were kind of connected by this little stream, and so basically they had the same level of water clarity and stuff like that. But what happened was, over time, conservationists had them sort of separate the ponds. Well, we can imagine that the pond that's in the mountain nearer to the pristine environment is going to sort of change in terms of its content. It's going to become more pure, and probably eventually sort of get rid of a lot of the pollutants and contaminants that were in it. Whereas the pond that's still near to the people in the industry might kind of be brown and mucky, and so we can see over time the two ponds would change, whereas before they were the same. And so basically that's how this works. If individuals are interbreeding between two populations, the two populations' allele frequencies are relatively the same. The second that they separate from each other and they're no longer breeding, those mutations and those changes are going to start to accrue, and they're going to change from one another. Okay, and so eventually this can lead to isolation barriers that make it impossible for gene flow to reoccur between them. And at the point at which gene flow can no longer occur between populations, that's where we say that they've become two different species, if we're using the biological species concept. I'm not going to go into the whole lumpers and splitters argument right now, but it is kind of interesting. I would definitely recommend, if you're interested in speciation, because it is fascinating, to read the book Speciation by Jerry Coyne, who is an alum of William & Mary. Sadly, I've never met him, but I love his books. Okay, not his blog. He's an angry person, but his books. Okay, so there are two main categories of reproductive isolation barriers. There's prezygotic and postzygotic. Okay, so prezygotic means before the organisms could even have a sperm and an egg try to fertilize with each other, it's not going to work. either their reproductive parts don't fit, or if the sperm was even able to enter into the other organism, it's never going to find its way to the egg. Even if it found its way to the egg, there'd be no way they could fuse because genetically they're just too distinct, too different. Post-zygotic means that after fertilization occurs, the offspring are either inviable or infertile. So basically it means that, yeah, the sperm and the egg fused, yeah, we had starting the development of the embryo, but because of their chromosomal differences, the embryo can't develop properly, or the embryo develops, but then and it grows into a healthy adult, but it's unable to actually breed itself. And when you can't have a species that can't breed, because all species must reproduce, and so at that case, we can't say that it's a species. And I think we talked about this when we talked about the mule as a mix of a horse and a donkey. So look at that example if you're wondering. Okay, so here we see with prezygotic barriers, we have temporal, behavioral, and geographic. Temporal basically means that the timing isn't right, so maybe one species breeds in the summer, the other breeds in the winter, and so even if they could breed together, they won't breed. Behavioral means that maybe their songs are different, or their mating dances are different, or maybe they just don't like each other, and so they don't breed. I mean, hey, that's relatable. And then there's geographic, right? So you could be just separated by huge distances, or mountains, or lakes, or rivers, or streams, or whatever, and make it impossible for them to breed together. Post-zygotic is going to be hybrid inviability. So basically when you have one organism is able, I got that confused in my head. So we were able to produce hybrids, but they basically die before they reach maturity. Then we have hybrid infertility, which is like the mule, and yes, the mule can grow to an adult size, but it can't reproduce. Then we have hybrid breakdown, where this one is really interesting. So yeah, the first hybrids are fertile, but then as they go on, as they reproduce and have more generations, each one becomes less and less fertile until they kind of are in the same boat as the mule, and then they all die. All of this happens because the chromosomes aren't playing nicely together. Okay, so whatever the cause of the reproductive barrier, whatever it is, whether it's post or prezygotic, that leads to speciation. And speciation is where we have new species. In other words, when the barriers for gene flow exist between populations such that they cannot interbreed. So, for example, the population of humans in my neighborhood cannot breed with the population of rabbits in our neighborhood, no matter how much people try. Hopefully they don't. And so basically, when we describe this process of speciation, we describe it in two different ways, whether it is allopatric or sympatric speciation. Allopatric speciation is when we diverge due to geographical isolation. So in other words, we are separated by distance or by some sort of physical barrier. Sympatric speciation means that we diverge within the same geographic location. So in other words, organisms are not constrained by physical barriers, but maybe by behavioral, by temporal, or maybe by the mechanical, which basically is the prezygotic, where your sperm and your egg can't fuse, your parts don't fit, whatever. And that's what sympatric is. So here's allopatric, right? It's an interesting photograph of allopatric. The mountains get in the way, and then sympatric speciation, a new species arises without that. Okay. And then we get to polyploidy. So polyploidy is fascinating. Polyploidy is essentially the idea that we're going to have the chromosome number of one organism is not going to be able to interact well with the chromosome number of another organism. Some organisms can actually have more than two sets of their chromosomes. This is really wild. So we know we're diploid, right? Typically a normal human cell is diploid because it has two of each chromosome. But some organisms can be triploid, which means they have three of each chromosome. Some organisms can be tetraploid, which means they have four, etc., on and on and on. It's very common in plants, happens all the time. Basically what happens is that in the process that's supposed to double the genetic information, it makes too much, there's a non-disjunction, or it just makes like twice as much as it needs to. So if sex cells fail to undergo cytokinesis, the chromosome number will double. Again, here we go. That's I should have read forward because that's what it was trying to say. And then these gametes will fuse with either normal or unreduced gametes, and this will result in polyploidy. And so if we imagine that a sperm has a diploid count of chromosomes and an egg has a diploid count of chromosomes, then 2 plus 2 is going to equal 4. That's a tetraploid offspring. If one organism has a diploid count, which is 2, and one organism has a haploid count, which is 1, add them together, that gives you a triploid, right? Now, polyploidy is, again, common in plants because they can self-fertilize. Wow, I really should have read the slide. They can self-fertilize, and and they can reproduce asexually. So both of these things are going to help them to be able to have polyploidy and also to be able to reproduce even if they're polyploid and there are no other polyploidies around. So in allium, we see that there's diploid garden onions, there's tetraploid garlic chives, and then there's octoploid blue chives, which is absolutely incredible. And so basically you can have all these changes now. If you have an even number of chromosomes, that can actually work. Those offspring often are fertile. They also, weirdly enough, have large fruits, mostly with the plants, so they'll produce the really large tomatoes or tetraploids and stuff. Triploids are infertile, usually. So triploids are usually infertile. They're seedless. That's where all of our seedless grapes and everything comes from. Basically, we have a diploid and a haploid fused together. Why the odd number of chromosomes and the even ones work differently, I'm not entirely sure, but I'm pretty sure that it just has to do with the number of chromosomes, the chromosome count. Because you need a certain number of alleles in order to create a trait, and it must be unbalanced in some sort of way. But I honestly don't really understand that as well as I should. Okay. All right, so what's the pace? How fast does speciation occur? I mean, if we look at all the species on Earth, we would, be tempted to say it occurs relatively quickly, but then you think about geologic time and how long it's taken for all these species to get here, so it usually takes a long time. But there's two general ideas behind this, which is phyletic gradualism, which basically means that speciation is a continuous slow change over time. So there's slow, gradual accumulation of mutations and alleles, and that slow process of reproductive isolation. And then there's the idea of punctuated equilibrium, which is basically that maybe a certain thing will evolve that causes a massive burst in diversity and activity, and that can lead to speciation very quickly. And so typically what we think of now is that usually it's gradual, and then there are periods of time where certain things will evolve that'll just spark a massive change, and then we'll get punctuated equilibrium and things will change very quickly. So here's an idea of phyletic gradualism, right? We can see that they kind of split off these butterflies and then slowly over time they become different. Whereas in punctuated equilibrium, we get the sudden change and then they basically remain relatively the same. So this is supported by many gaps in the fossil record. Of course, those gaps might exist because of geological disturbance and stuff like that. And this is, of course, supported by all the intermediate fossils in the fossil record. So we're kind of stuck between these two. We think there's probably a blend of the two is true. And then we have this here. So the speciation is, again, sort of a tricky business. Not everybody agrees on what constitutes a species. There are certain organisms that don't reproduce sexually, for instance. How exactly do you say that they are the same species or not? On the biological species concept, we base it off of whether or not they can reproduce. But if they don't reproduce sexually, I mean, what do we say? We might be unable to determine the breeding capacity of ancient organisms through fossilized remains. So, we're not really sure whether or not we can say that certain fossils that we find are different species than certain other fossils. Of course, it might physically be impossible for certain members of a species to mate, but maybe their sperm and their egg could fuse. It's possible, so we're not really sure. And then, of course, there are ring species, which is pretty interesting. It's basically where you have, and this has happened with a couple of birds across the U.S. and lizards on different islands. You have one population, and then they change and diversify over time. They create this population, it speciates from this one, this population speciates from this one, and eventually they kind of loop back around where the end result of all this speciation in these different populations is right next to the original population, and they can no longer interbreed. So really interesting. Which is, just incredible, but A and B could breed, B and C could breed, C and D could breed, D and E could breed, but A and E couldn't breed. It's a really fascinating situation. But anyway, so hopefully you guys understand now speciation. You feel relatively comfortable with it. Sorry, this one was a bit long. I'm really trying to get down to about 25 minutes for these videos, or 20 minutes. So there's just a lot to cover today, and want to make sure I went over everything. Hope you guys are having an awesome day, and I miss you guys. I hope you're doing well, and I can't wait to see you on the Google Meet on Friday if you're able to attend. All right, guys, take care of yourselves.